I'll be talking about how the COVID-19 vaccine works. This is the SARS-CoV-2 virus, the virus causing the current pandemic. If you look at the virus closely, you might have noticed these spikes around the virus. These spikes are called spike proteins. They're kind of like the keys for the virus to enter the cell. Now, you can't open all doors with a key. You can only open doors that have the right lock for the key to fit in. The same goes for the virus. The SARS-CoV-2 virus can only enter cells with a receptor called the ACE2 receptor. The receptor is kind of like a door for the virus to enter the cell. The SARS-CoV-2 virus also has a lipid bilayer envelope around it. Now, this doesn't mean it's inside an envelope. The envelope is like an extra layer of the SARS-CoV-2 virus that not all viruses have. Then we have this capsid, the layer right below the envelope. All viruses have a capsid, but not an envelope. The capsid is the layer that surrounds the genetic material of the virus. For the SARS-CoV-2, this genetic material is called RNA, or ribonucleic acid. But the genetic material of a virus can also be DNA, deoxyribonucleic acid. The main goal for every virus is to reproduce. However, viruses don't really have the proper machinery necessary for the process of viral replication. They need a cell with the proper machinery to do it for them. To tell the cell what exactly it must do to produce more viruses, the virus will give its RNA to the cell. The cell is then going to blindly read the RNA and do as it says. RNA is coded in 4 molecules: adenine, uracil, cytosine, and guanine. The cell is going to read the RNA in codons. A codon is basically a 3-letter pair. For example, AUG would be a codon, AGC would be a codon, and so forth. Now, each of these codons will be assigned an amino acid. In the case of AGC, it will be serine. The SARS-CoV-2 has about 10,000 codons in its RNA. Now let's get straight to the point. How does the COVID-19 vaccine work? Well, the COVID-19 vaccine is basically some mRNA, an abbreviation for messenger RNA. Now, before, when we were mentioning RNA, we were talking about mRNA, since mRNA is the only type of RNA that can be read by cells to produce some type of proteins. mRNA is kind of like a messaging system between cells. For example, if one cell is infected by a virus and figures it out, it will send some mRNA which will produce a protein called interferons, which will alert all the other cells to get into fighting mode. So the cell is going to think that the mRNA from the vaccine is coming from another cell. So it's going to read it. But how? Well, to read the mRNA, the cell goes through a process called translation. So basically, we have this little organelle, the ribosome, in the cell. The ribosome can't really read the mRNA though. It needs the help of the tRNA, an abbreviation for transfer RNA. So let's go closer into the tRNA. As we can see, the tRNA contains what we call anticodons and the amino acids that match to the codons that the anticodons bond to. A tRNA will bond to its matching codon. This will let the ribosome know that it must take the amino acid off the tRNA and put it in a chain. Then, let's say another tRNA comes. Then the ribosome will take this amino acid on the tRNA and put it in the chain. Here, as you can see, we have a small peptide, which is just a small chain of amino acids. But in the case of the vaccine, we'll have a much more complex peptide known as a protein. The mRNA of the vaccine will make the ribosomes produce the spike protein of the SARS-CoV-2 virus. If you remember, the spike protein are the proteins outside the SARS-CoV-2 virus that lets the virus get inside the cell. Okay, so now that the cell read the mRNA and got some spike protein, what is it going to do? Well, the cell is going to identify the spike protein as foreign. Since this is only a spike protein and not a full virus, it's not going to be able to escape the cell's defenses. The cell is going to break it down into pieces. Then, it is going to alert the immune system by displaying one of the pieces on an MHC1 protein. This is going to alert a killer T cell, also known as a cytotoxic T cell, that the cell is now a virus factory. We don't want any viruses to be created in the body, so the killer T cell is going to kill the cell using something we call cytotoxin. When the killer T cell does this, most of the spike proteins will be broken down. But some of them might just escape. So now what will happen to these proteins? Well, these proteins will either be found by some nearby macrophage or attached to another cell. A macrophage is a type of immune cell. Macrophages are part of a group of immune cells called phagocytes. When a phagocyte sees a virus or a part of a virus, like this spike protein, it will engulf or eat it in a process known as phagocytosis. Then an organelle of the macrophage, which we call the proteasome, will break down the spike protein into small parts. Then, instead of displaying one of these small parts on the MHC1 protein, it will display the piece on the MHC2 protein. Now, this can activate a certain helper T cell, a type of immune cell. This helper T cell must have the right receptor to bond to the MHC2 protein. Once the right helper T cell has been activated, it will call another type of immune cell called a B cell. Each B cell has a specific type of antibody. These antibodies are made to tag and disable pathogens like viruses and bacteria for macrophages to attack. In the body, there will be at least one B cell that has the right antibodies to attach to the spike protein of the SARS-CoV-2 virus. Let's say that this B cell is the one that has the right antibodies and can attach to the part of a spike protein displayed by the cell. This means that if the SARS-CoV-2 comes, then the B cell will be able to use its antibodies to disable the virus so it can't do anything. Well, then the T cell will activate the B cell so it produces two other types of B cells, effector B cells and memory B cells. Memory B cells will memorize the antibody in case of another attack by the SARS-CoV-2 virus happens. The effector B cell will start mass producing the antibody so the body can be ready to fight off the virus. Now we have talked about what will happen if the remaining spike protein goes next to a nearby macrophage. But what if they go to just another cell? Well, then inflammation could occur. These ACE2 receptors are not just receptors. They carry something we call angiotensin. Angiotensin is necessary for homeostasis across the body. If these ACE2 receptors are blocked by spike protein, then they can't be used for these processes anymore. And this is why some people can get inflammation after taking the vaccine. So let's say that you figure out that you need 2 doses of the vaccine, and you only took 1. Now you can't help but wonder, why do you need to take another dose? Think of it like this. The first dose pushes you this much closer to making you immune. But the second dose is actually the dose which uses the push given by the first dose to get you much closer to making you immune. It doesn't actually make you immune because the vaccine isn't 100% effective, but it does dramatically decrease your chances of getting the virus. Thank you for watching. I hope you enjoyed this video. You can watch our video on 2 reasons why COVID-19 is so deadly, and you can watch our video on what are viruses. Please subscribe to Kids Teach Kids and stay tuned. Bye!